Welcome back, everyone. Okay, let's dive into this because today we're exploring something that genuinely sounds like it was ripped straight out of a sci-fi movie. But I promise you, this is an emerging, hard science reality. Imagine just for a second building a computer, but instead of silicon chips and copper wires, you're building it out of actual living human brain cells. Wild, right? We're talking about a totally new frontier in biocomputing, and the official name for it is organoid intelligence. It's a real game changer that promises to completely flip how we think about computing power, memory, and honestly, even the biological nuts and bolts of learning itself.
So why on earth are scientists going down this biological route? Well, it all comes down to two numbers: 21 megawatts versus 20 watts. 21 megawatts is the staggering, mind-boggling amount of power it takes to run the world's most powerful supercomputer. I mean, that is literally enough energy to power an entire small town. On the flip side, 20 watts. That's the hyper-efficient, incredibly tiny amount of power needed to run a human brain. Just a dim light bulb's worth of energy. This massive contrast right here is the absolute foundation of the whole problem we're trying to solve today.
Which brings us to Section 1, the AI power problem, the unsustainable energy cost of modern computing.
Look, modern AI is absolutely incredible, right? It's revolutionizing basically every industry. But it has a massive fundamental limitation when it comes to data and energy, especially as we try to get it to mimic biological processes. And this brilliantly illustrates exactly what we're up against. If you compare the Frontier supercomputer, which is the world's first exascale supercomputer, to the human brain, their raw processing speed is actually kind of similar. They both operate right around the 1 exaflop mark. But how they actually pull that off? Totally different stories. The supercomputer needs that 21 megawatts of power we just talked about. It takes up thousands of square feet with miles and miles of cabling. Meanwhile, your brain weighs about 3 pounds and again, runs on just 10 to 20 watts. We are talking about human biology operating with 1,000,000 times better power efficiency than our absolute best machines.
And it goes beyond just the power needed to run the hardware. There's also this massive data efficiency problem in how machines learn versus how biology learns. Take AlphaGo. That's the famous AI that beat the world champion at the complex game of Go. To train that AI, it needed to process 160,000 games. To put that into perspective, a human would have to play continuously for over 175 years to get that kind of experience. Actually, get this. The energy required just to train AlphaGo could sustain the metabolism of an active adult human for an entire decade. Now contrast that with biological brains. Whether it's a human or a much simpler organism, we can learn complex tasks with sometimes less than 10 to 100 tries. Modern AI just heavily relies on massive, unsustainable amounts of repetitive data and energy that biological brains simply do not need.
So let's get into Section 2. Enter organoid intelligence. Intelligence in a dish.
Because the energy consumption for machine learning is skyrocketing to totally unsustainable levels, researchers are pivoting. Instead of trying to force computers to be more brain-like, they're exploring a completely paradigm-shifting solution, harnessing actual human brain cells to do the computing. So the crucial point is establishing exactly what we're talking about here. Organoid intelligence, or OI for short, is an emerging multidisciplinary field. The goal is to develop biological computing using 3D cultures of human brain cells, which scientists call brain organoids, and combine them with brain-machine interface technologies. Basically, it's about leveraging the self-assembling machinery of biological neural networks to process and remember information. Now, you might be wondering, why 3D brain organoids? Well, for the longest time, scientists use these flat 2D cell cultures in petri dishes, but those do a pretty terrible job of replicating the real, complex environment of an actual brain. These advanced 3D organoids are vastly superior. For one, they have a really high cell density, much closer to a living brain. They also show spontaneous electrophysiological activity. That means they're actively firing off electrical signals all on their own. And amazingly, the axons in these organoids show up to 40% myelination. Myelin is essentially the insulation around the electrical wires in your brain, and having it can boost conductivity like 100-fold. Plus, these 3D structures are enriched with glial cells, which we now know are absolutely crucial for pruning synapses and forming memories. They are literally the perfect candidates for actual biological computing.
Which brings us to Section 3, Engineering the Biocomputer, how to build a living machine. Okay, so it's one thing to have a tiny cluster of brain cells sitting in a lab, right? But it's an entirely different beast to keep them alive long-term and figure out how to communicate with them to build a working machine. To make OI a reality, researchers have to nail 3 major bioengineering steps. Step 1 is all about scaling up. Right now, your typical organoid might only have around 100,000 cells.
Scientists need to bump that up to about 10 million cells to get the kind of complexity required for computing. Step 2 is connecting them to 3D microfluidic perfusion systems for life support. And then step 3 is building the interface, specifically using 3D microelectrode arrays to handle transferring data in and out of the living tissue.
Let's zoom in on step 2 for a second. As an organoid gets bigger, the cells stuck right in the middle will literally starve to death if they just rely on passive diffusion for their food. 3D microfluidic perfusion systems completely solve this roadblock. You can basically think of them as artificial, synthetic blood vessels. They don't just deliver the essential oxygen and nutrients needed to keep the whole organoid alive and healthy, they also let scientists send highly targeted chemical signals, like neurotransmitters, to very specific parts of the organoid at exact times.
And then for step 3, the data transfer, we have 3D microelectrode arrays. Here's the thing, traditional flat electrode chips don't really work when you're dealing with a spherical organoid. So researchers engineered this ultra-soft, flexible shell that actually folds itself right around the organoid. It's essentially like fitting a tiny, highly advanced EEG cap onto a ball of cells. This lets scientists read the spontaneous electrical signals coming off the entire surface, and this is the crazy part, it lets them write information back into the organoid using electrical stimulation.
So, once you've got the life support and the data interface all hooked up, how do you actually train this thing? Well, in a pretty brilliant twist of irony, we actually have to use artificial intelligence to train the biological intelligence. The organoid generates these massive, super complex datasets of electrical and chemical responses. We need AI and machine learning algorithms to decode all those signals and map out the relationship between what goes in and what comes out. By continuously feeding that biofeedback back to the organoid in a closed loop, researchers can stimulate synaptic plasticity, and that right there is the fundamental biological mechanism for how we learn and form memories.
Let's move to Section 4, Transforming Medical Research, and see how this builds into something far beyond just computing. From computing to curing.
Developing OI isn't just about building a faster, greener computer. It's really about shifting the whole narrative from computing to curing. By bioengineering these complex, working models of the human brain, we are essentially creating the very first fully human-based preclinical models for medical research, and that has absolute game-changing implications for some devastating conditions. Think about Alzheimer's disease and other dementias. A lot of clinical trials fail because animal models just don't accurately mirror what happens in a human brain. The same goes for neurodevelopmental conditions like autism spectrum disorder, schizophrenia, and rare white matter diseases like leukodystrophies. Here's how that actually works in practice. Because these organoids are grown from stem cells, which can be safely sourced from something as simple as an adult skin sample, researchers can grow brain organoids using cells donated by people actually living with these conditions. So, by comparing an organoid grown from an Alzheimer's patient with a healthy control organoid, scientists can study the exact physical and genetic mechanisms causing cognitive decline. And even better, they can safely screen potential new drugs or test for toxic side effects directly on human tissue before that drug ever goes anywhere near a living patient.
Finally, we arrive at Section 5, the ethics of intelligence, consciousness, sentience, and responsibility.
We absolutely cannot talk about creating living, learning, neurological networks without addressing the profound philosophical and ethical implications of this work. I mean, it's the elephant in the room, right? The critical question that naturally pops up is this. As we scale these organoids up, as they get more structurally complex, and as they start building primitive memories and learning from their environment, could they exhibit consciousness? Could a lab-grown brain organoid actually feel pain or experience suffering?
To tackle that, scientists and ethicists draw a really vital distinction between sentience and consciousness. Sentience is just a basic responsiveness to sensory input. Think of a really simple organism reacting to a flash of light or chemical. Consciousness, on the other hand, is a much higher order capacity. It implies being broadly aware of your surroundings and having a true, lived phenomenological experience. Now, what's really interesting about this slide is that it clarifies a major misconception. The researchers behind organoid intelligence are strictly aiming to achieve basic sentience for the sake of computing, just a simple stimulus-response loop. They are absolutely not trying to create human-level consciousness in a dish.
But trust me, researchers aren't taking any of this lightly. They're dealing with something known as the Greeley dilemma, which is basically where every incremental success in modeling the human brain raises the exact same ethical concerns that made you want to build the model in the first place. To handle this responsibly, they use an embedded ethics approach. This means expert ethicists aren't just, observing from an ivory tower. Actually, scratch that, it's more like they are right there in the trenches, working alongside the scientists in the lab, identifying and hashing out ethical issues as the research happens in real time. It's this continuous, three-way feedback loop between researchers, ethicists, and the public to ensure this paradigm-shifting technology develops responsibly.
We are quite literally at the dawn of an awe-inspiring technological leap. By marrying the unmatched power, efficiency, and learning capacity of biology with advanced microfluidics and AI, the biocomputer is moving straight from science fiction into laboratory fact. So, I want to leave you with this one provocative thought. If we actually pull this off, if we succeed in building intelligence in a dish, solving the AI power problem, and unlocking all these new medical treatments, what will creating biological computers ultimately teach us about the true nature of our own minds? Thank you so much for joining me on this explainer. Keep learning, keep questioning, and I will catch you on the next one.